Now let's talk about how acquired immunity develops memory against specific pathogens. This is also possible due to the cell-mediated response. As a result of T lymphocytes recognizing epitopes of protein antigens during cell-mediated immunity, that is during clonal selection, numerous circulating T8 memory cells and T4 memory cells develop which possess anamnestic response or memory. These T memory cells persist for the remainder of a person's life. As we have discussed before, that a T cell persists as an effector T cell until its fate is decided. Similarly, effector memory T cells circulate in the blood, whereas tissue-resident memory T cells are found within the epithelium of the skin and mucous membranes. CD8 TRM cells are typically activated by viral antigens and subsequently produce inflammatory cytokines that trigger an innate immune response for nonspecific antiviral activity. CD4 TRM cells are found in clusters surrounding macrophages in the mucosa. Unlike TEM cells, TRM cells do not circulate in the blood and are not replenished from the blood. They remain in peripheral tissues. Subsequent exposure to that same antigen results in a more rapid and longer production of cytotoxic T lymphocytes, a more rapid and longer production of T4 lymphocytes, and triggering of nonspecific innate immune responses. As a result, the immune response of a person improves with repeated exposure, as the invading pathogen is instantly recognized by the memory cells and a cell-mediated response is triggered rapidly. The triggering of the innate response also helps greatly in the host's defense. Cellular immunity is also the mechanism behind delayed hypersensitivity. Hypersensitivity reaction are of 4 types. Type 1, anaphylactic or immediate. Type 2, cytotoxic or antibody-mediated. Type 3, immune complex-mediated. Type 4, delayed or T-cell-mediated. Type 1, 2, and 3 are antibody-mediated, whereas Type 4 is a delayed reaction brought about by T lymphocytes. Delayed hypersensitivity is generally used to refer to the harmful effects of cell-mediated immunity. It is a delayed reaction because it takes time and starts in hours or days after contact, unlike those that are antibody-mediated. Some of the biological implications of the delayed hypersensitivity are tissue and transplant rejections. The success of organ transplants is, in large part, determined by the compatibility of the MHC genes of the donor and recipient, as well as the cell-mediated response of the recipient. Contact dermatitis, type of eczema triggered by contact with a particular substance, such as poison ivy. Positive skin tests like the PPD test for tuberculosis after the symptoms of the illness have subsided. Granuloma formation during tuberculosis. Destruction of virus-infected cells is also part of the delayed hypersensitivity reaction. That is all about the cell-mediated immunity. Stay tuned to sqadia.com.